Multiple myeloma is a blood cancer that affects the bone, and it causes terrible complications.
The problem of this disease is even with the most aggressive treatment that we use today, 5 years following treatment, only 30% of the patients will survive.
Although there are so many new treatments being developed for the treatment of myeloma, there is still as yet no cure. And that really is the goal of our work, is to try and find a cure for this devastating disease. What we think is happening when patients go into remission is that there remains within their body a few cancer cells that are dormant or sleeping.
And they can sit in the bone in very specialised environments for long periods of time. And then something wakes up those cells, and when they get woken up, they form overt tumours.
So really, to understand what causes relapse and to really try and find a cure for myeloma, we think we really need to get a handle on the biology of these dormant cancer cells.
What we're trying to do together is really use these very strong single-cell approaches to characterize these stem cells and understand new ways to actually treat them. And once you eliminate the stem cells, the treatment will be much more effective and the disease will not come back.
So one of the big challenges has been to find these sleeping cells in the skeleton. And you can imagine they're hidden away in very discrete sites in bone. So actually finding them has been a major challenge.
We had to develop special techniques and technologies that would allow us to perform microscopy in a living mouse and watch the myeloma cell. We could see these myeloma cells, we could see the dormant ones and where they reside in the bone. But the next question for us was to understand why they were doing this.
And this coincided with work from Amit's group at the Weizmann Institute, who were pioneering the development of single-cell sequencing technologies.
With Peter, what we're trying to do is identify what are these stem cells. We've been developing very sophisticated technologies that allow us to measure every single RNA within the cell. And that really gives us a complete information of each individual cell.
Because these cells express very different genes, this gives us a really unique opportunity to think about targeting those cells specifically. And if we can eradicate those cells, we stand a chance of preventing disease relapse and making progress towards curing myeloma. It's been a great opportunity to work with Ido and his colleagues at the Weizmann Institute. I think our individual research experiences are very, very complementary, and it's really allowed us to move this project forward much, much more quickly than we would have done elsewhere.
Combining our vision and technologies with Peter's long-standing research on this topic can make a huge impact.